Good afternoon, everybody. Thank you so much for your patience. We were having a few technical issues. I'm Kyra Mann, CEO of MitoAction. Welcome to today's monthly expert series presentation. We're excited to have members of the PTC Therapeutics team with us today to share about their current trial for PTC743. Before we get started, just a few housekeeping items. Today's presentation will be recorded and available on the MitoAction website in the coming days, as well as on our podcasts on Apple Podcasts, Google Play, and Spotify. If you're joining us via phone, I would encourage you to follow along with the presentation slides that can be found on our website under the monthly expert series. If you're joining us via your computer, you should see the presentation on your screen. We encourage you to ask questions throughout the presentation using the Q&A feature at the bottom of your screen on the menu bar. If you're calling in via phone, feel free to submit your questions to us by email at info@mitoaction.org. We will do our best to get through as many questions as possible at the end of today's presentation. Our speakers for today's presentation are Dr. Matt Klein, Chief Development Officer, and his colleague, Dr. Francesco Bibbiani, Vice President of Clinical Development at PTC Therapeutics. Prior to joining PTC Therapeutics, Dr. Klein was CEO and Chief Medical Officer of BioElectron Technology Corporation, a biotechnology company focused on development of redox active small molecules for mitochondrial disease and related disorders of oxidative stress. Prior to this role, Dr. Klein was the Otho Washington Research Foundation Chair of Restorative Burn Surgery at the University of Washington. He completed his undergraduate degree at the University of Pennsylvania, where he graduated summa cum laude and Phi Beta Kappa and received his MD degree with honors from Yale University. Dr. Francesco Bibbiani is a board-certified neurologist with over 20 years experience in clinical development between the pharmaceutical industry and the experimental therapeutic branch of the National Institute of Health, where he conducted proof of concept trials in several neurological indications. In his past positions, Francesco conducted various clinical trials in Alzheimer's disease, epilepsy, and liver disease, all studies that led to multiple regulatory submissions and approvals with the FDA, EMA, and other global regulatory agencies. Currently, in his role as Vice President of Clinical Development at PTC Therapeutics, he is the clinical lead of the DMD and mitochondrial epilepsy projects. He received his MD with honors from the University of Pisa, Italy, where he also completed his residency program in neurology with honors. Please join me in welcoming Dr. Matt Klein and Dr. Francesco Bibbiani.
Thank you very much, Kyra, and thank you to everyone who's joined us here today. As always, I'm incredibly grateful to have this opportunity to speak with you all, and really, really excited to be sharing the great news that we are initiating the newest 743 mitochondrial disease trial. I am here today with my colleague, Dr. Francesco Bibbiani, who will be leading the 743 trial. But before turning things over to Francesco, I wanted to just start with a few introductory comments. First, as many of you know, EPI 743 is now PTC 743. BioElectron's assets, including a drug candidates, were acquired last year by PTC Therapeutics. And so now 743 and the majority of the BioElectron team are now part of the larger PTC family. PTC is a rare disease-focused global biotechnology company with headquarters in New Jersey and offices worldwide. PTC was founded 22 years ago, and with the singular focus of developing drugs for patients with rare disease. We've kept that mission, and that remains our focus, and we now have a number of drugs on the market for rare diseases, as well as a number of drugs in development for a number of different rare diseases. The acquisition of BioElectron and the transition of EPI 743 to PTC is really great news for the mitochondrial disease community. PTC brings added muscle and added experience to the development of 743 and other compounds for mitochondrial disease. And the first of these efforts of bringing things forward for mitochondrial disease is, of course, this trial in mitochondrial epilepsy. On this next slide, this is the summary of all of the different programs in PTC. We have a number of different drug development programs from a number of different platforms. We have our approved therapies for Duchenne mitochondrial dystrophy. We have other products that we're commercializing in Latin America. We also have a program in splicing and a recently approved drug for spinal muscular atrophy that was developed with Roche. We have gene therapy programs with our lead program in AADC deficiency, for which we're in the process of preparing for regulatory submissions for approval in the EU as well as the US. We have programs in metabolic disease and oncology. And of course, our MitoE platform with our programs being developed for mitochondrial disease, including this trial in Mito-Epilepsy, as well as Friedreich ataxia, and a number of different compounds in the pipeline being developed for a number of different CNS diseases, as well as mitochondrial disease. The focus of today's presentation, of course, is to provide you more information on this PTC743 mitochondrial epilepsy trial, which has already initiated in the United States and will be run at study sites not only in the US, but worldwide. So I'll turn the presentation now over to Francesco, who will give you a number of additional details on the trial. Francesco?
Yes, thank you, Matt. Thanks everybody for being part of this presentation and attending this presentation. As Matt said, we are very proud of the study and very excited to continue the development of the drug. So just a brief introduction on Vatiquinone, PTC743. It is a vitamin E derivative. More precisely, it's the oxidation product of alpha-tocotrienol, which is one of the E vitamins. And the action of Vatiquinone is on the enzyme 15-lipoxygenase. The role of the enzyme is to transform polyunsaturated fatty acids in response to oxidative stress to oxidative fatty acids, and that increases inflammation and oxidative stress. In presence of Vatiquinone, Vatiquinone inactivates this enzyme, therefore resulting in decreased inflammation and oxidative stress and improving cellular viability. In the time between 2009 and 2012, there was an expanded access program that was run that enrolled about 94 patients that were within 90 days of the end of life care. This was initially a 13-week treatment, followed by a long-term extension phase. The study measured the mortality, the drug safety, and the pharmacokinetics of the drug. In terms of results, of the 94 patients that were enrolled, 42 are still currently alive and on treatment. The mean survival is over 4 years, and considering that these patients were within 90 days of end of life care, it is a very important achievement. Safety-wise, 743 was safe and well tolerated, and the clinical efficacy findings included reduced seizures, improved neurological and neuromuscular function, a reduction of status epilepticus, and also hospitalization due to the disease itself. Also improved CNS biomarker across the multiple disease subtypes. In terms of the treatment, these are the examples of the cases that were treated. Overall, we see a decreased seizure frequency, an improvement in the status epilepticus occurrence and the severity, the reduction of hospital-related occurrences, disease-related hospitalization, as we said. All this throughout the different subtypes of the disease. So consistency of these outcomes within all the mitochondrial epilepsy syndromes. In terms of a study that was run in Leigh syndrome, it was run in several countries, in Italy and in US, and we saw that the baseline seizure frequency went from either mild or moderate to mild or completely absence of seizures. In terms of the study itself, so why are we running a study? So first of all, because as you know, mitochondrial epilepsy, mitochondrial disease is often associated with epilepsy. About 40% of patients have also concomitant epilepsy. This type of seizures are typically difficult to treat. They are refractory to the traditional anti-epileptic drugs. Then the drug has both a scientific and a clinical rationale for treating this disease. So it responds to animal and in vivo and in vitro models of refractory epilepsy, and also in clinical with the experience that we just went through. The study has as the primary endpoint the percent change in baseline seizure frequency as observed in a 20-day period and measured by seizure diaries. The key secondary endpoints are the number of disease-related hospital days and the occurrence or recurrence of status epilepticus. Also, the study will evaluate safety in these patients. The design of the study is a double-blind placebo control that has a screening phase, which will run for approximately 42 days, followed by the placebo control phase for 24 weeks. And then after the 24 weeks, the subjects will have the opportunity to continue in a long-term extension phase of the duration of 48 weeks, after which they will be followed up after drug discontinuation. The reason why we have a long screening and run-in period is that because during this time, the patients and their caregivers will get familiar with the main instrument for the measure of seizures, which is the seizure diaries. They will fill out the diary, recording their seizures, and once they feel that are familiar enough and they have recorded the seizures accurately, they will enter what is called the run-in phase, in which they will measure the seizures over 28 days, which will become the baseline seizure count. In order to qualify for the study, subjects have to be less than 18 years of age, have a caregiver that is able and willing to complete seizure diaries for the duration of the study. It's a hard task. I mean, if you think about it, it's a daily task that they have to complete the seizure diaries. Although with the electronic seizure diaries, it's a fairly simple task. They have to have a genetic confirmation of an inherited mitochondrial disease with associated epilepsy. That includes Alpers-PLG, Leigh syndrome, MELAS, and other genetically confirmed mitochondrial disease, secondary to a genetic mutation. They have to have seizure despite being on at least 2 anti-epileptic drugs, and have enough seizure to actually measure a therapeutic effect. So they have to have at least 6 seizures, motor seizures, during the first 28 days of the baseline period. We chose motor seizures because they're easier to identify and to track. They also have to have 2 of the motor seizures in the first half of the 28 days and 2 in the second half, so that we don't have periods that are without seizures, so that depending on where you measure the 28 days, you might have a confounding factor. So we want the seizures to be relatively spread out equally in these 28 days. For the same reason, we want them not to have 20 days of seizure-free period. Also, they have to be able to fill out the diaries fairly consistently and have at least 80% seizure diary entered at each time. We will document the medical history, and including the associated concomitant treatment for at least 6 months before screening. And they have to be agreeing not to enroll in any other non-approved treatments for at least 30 days prior to the baseline and during the duration of the study. As we said, they have to be on a stable regimen of anti-epileptic drugs, 60 days prior to baseline visits, so no adjustments are allowed right before entering the study. Otherwise, they could confound the baseline number of seizures unless they're really stable on their ongoing regimen. Same can be said for dietary supplements, or if on a ketogenic diet, being stable for at least 90 days, since we know that the effect, to reach maximum effect for ketogenic diets, it might take up to 90 days. Therefore, we want them to be stabilized from that point of view as well. So we need an electroencephalogram in EEG, at screening, or even a historical EEG for diagnostic confirmation of seizures. That doesn't mean that we need to record a seizure in the EEG, but we just need to have an EEG that is compatible with the seizure, with an epileptic syndrome. So the Seizure Diaries, I mentioned before, it is the key instrument for the study because it's, first of all, our primary endpoint, but also because the accuracy of the measurement, especially at the baseline, is absolutely paramount, because everything is then going to be compared to that. So as we said, we have these conditions of having seizures that are relatively spread out during the 20-day period, as we described before. In terms of randomization, the subject will be randomized 1-to-1 to Vatiquinone or placebo. So subject will have the same chance of receiving either placebo or Vatiquinone for the first 28 weeks, after which they will switch to an open label phase where everybody will receive Vatiquinone. Not the patients or the physicians or ourselves will ever know which one of the patients were on what drug during the double-blind phase. Subjects will be also randomized to the different types, or at least the main types of mitochondrial diseases. So the Alpes-Paul-G, Leigh syndrome, MELAS, they will have at least 6 patients in each of these 3 categories, so that we could have a fair representation of at least the most common of these diseases in our study. So the maximum duration will be approximately 72 weeks, including the placebo control and the extension and the screening phase. The number of sites. So we want to have at least 12 sites globally, but that are randomizing. Sometimes you activate the sites and they're not screening. So, we want to have more sites. Even more so in these times of uncertain situation where site closures is possible due to the pandemic, and therefore we are working with a larger number of sites that are also distributed geographically in different areas, so to be able to maintain the study enrolling in any event. One thing that I wanted also to point out is that the study has been planned with also the opportunity to do visits remotely for the same reason, and do like telemedicine and remote visit if needed, in case it will become necessary. We want to enroll approximately 60 subjects, and more sites and more subjects might be added if necessary. So that concludes my presentation. I guess we're available for questions.
Okay, thank you both so much. So I have some questions that have come in that we'll go through. Can you talk a little bit about the age limitations and why it's being limited to patients who are 18 years old, younger than 18 years old, and is there a minimum age requirement as well?
So a lot of these diseases affect primarily younger patients. We do not have a minimum age, so anybody below age of 18 is allowed to enroll. That's where we feel like the vast majority of our patients will be as a first trial in this indication.
Yeah, and I'll add to that, is of course, while we're aware that there are many adults with mitochondrial disease, of course, and that they also have seizures and may also benefit from 743. With all the work we've done over the years, one of the key things we've learned in doing trials of mitochondrial disease is try to minimize heterogeneity. We know that that's a major challenge in mitochondrial disease trials, and age is one of those factors where we can try to minimize heterogeneity. And so we view this trial not as the trial, the only trial, but as, again, a first trial in mitochondrial epilepsy with, again, a very strong desire to, at long last, advance 743 for approval, with the ultimate goal of trying to make 743 available to as many patients who may benefit as possible.
Perfect. Thank you so much. I appreciate that. So with a genetic diagnostic, genetic confirmation being required, are there opportunities for genetic testing for a patient that presents with one of these diagnoses that has not yet received it, but it seems very likely?
So we do not confirm the genetic nature of the disease. We don't repeat a genetic testing in the study. Usually these patients are known to their sites and have been already tested. If not, if they haven't been tested, there might be opportunities for the site to do so at the time. Yeah.
Okay. You talked about the other treatment regimens and dietary plans. Does a participant have to go off of any medications or supplements in order to participate in the trial?
No, they have to be on a stable regimen of it. So for pharmaceuticals, for any other anti-epileptic drug, we require them to be on a stable dose for at least 30 days. So the slide still says 60, but we did change it in the last amendment to 30 days. And the same thing is for supplements. So as long as it's stable, we do not require any change. And we also expect that unless there's a safety issue, that this treatment will be kept stable, at least in the placebo-controlled phase. This is all to avoid any confounding factors of drug that have been increased, that will be increased during the efficacy portion of the study, or reduced or changed. We want to maintain the situation as stable as possible so that we can appreciate the effect of Vatiquinone on the seizures.
Perfect, thank you. Are there any implications of this study for those who have mitochondrial disease that don't have epileptic events, or perhaps potential for considering this down the road?
I'm sorry, I don't think I heard the
question. I think what they're asking is for those who have mitochondrial disease but are not experiencing epileptic events, is there any consideration or potential consideration down the line that this could also be a potential treatment for non-epileptic patients?
Right, so maybe I'll let you, Matt, answer that.
Yeah,
not in the study.
Yeah, absolutely. I mean, as I think people are well aware, 743 has been studied in a number of different mitochondrial disease subtypes. We've collected a lot of strong data showing clinical effect over the years, and importantly, safety in both children and adults. Our vision is to try to get 743 available to as many patients with mitochondrial disease as possible. But when we put our drug developer hats on, one of the things we need to do is come up with a way to gradually get it approved for more and more patients. And we view this trial in mitochondrial epilepsy, or patients with mitochondrial disease and seizures, one that's based on what's known about the science of the drug, the science of mitochondrial disease, data we've collected in a number of studies, is an appropriate first indication for approval, again, with the goal of trying to make the drug more broadly available to those adults as well as children, patients with seizures and without seizures. It's just a matter of a process of moving forward one by one.
Right, so step one in a long process, right?
Probably more like step 8 in a long process. Yeah.
Absolutely. So for a patient that is, is listening to this, who hears you describing them as this ideal patient or their child, what are the steps that you would recommend to them to connect with somebody if they're not being seen by a mito specialist, in order to connect with a center for more information about the trial?
So the study is listed in clinicaltrial.gov, like all the studies that are ongoing worldwide. Clinicaltrial.gov also lists the sites that are currently participating in our study. Also, we do have a website dedicated to the study that lists contact information and emails information and phone numbers that they can reach to know more about the study. And that is also included in the PTC website, PTC Therapeutics website.
And it's also important that even if your mitochondrial doctor and the place where you go for care isn't a study site, you're still eligible for the study. In fact, we've already had patients who are coming across a number of different states to participate in the study, and that's something we're aware of and make available. So everyone is welcome if you feel you meet the criteria.
Okay, great. You talk about the ideal number is registering 60 patients. Does that mean the study will be limited to 60, or that's what your goal for the minimum is?
That is the goal. It's usually usually we stick to the projected number. That's the minimum number that we would like to have in order to have enough power to detect the difference. It's always approximately because we can go above a little bit, but this is the number that we're targeting.
Okay, great. Someone is asking about disease prevalence in the US. Do you have any insight to what that patient population is in terms of, patients who have 18 and under, who have mitochondrial disease, who are suffering from epilepsy? You talked a little bit about those statistics in the presentation.
Francesco, I can grab that. I mean, we've done a lot of work looking at this, and the number that we have calculated based on literature and a lot of other work we've done in our teams is that if you look at the US and Europe and probably Latin America, places where we're looking to do the study, we estimate that there's probably around somewhere between 11 and 15,000 patients in those areas. So obviously a portion of that would be the United States, a portion of that would be Europe, a portion of that would be Latin America. But what we do know, there's been a lot of really great work done by Shamima Rahman and from London, Russ Senato in Seattle, and other mitochondrial doctors around the world. We know that somewhere between 40 and 50% of all patients with mitochondrial disease have seizures. And we also know that probably 90% or more of patients with mitochondrial disease and seizures don't fully respond to traditional seizure medications. And the reason for that is traditional seizure medications don't target the energy pathways that lead to seizures in patients with mitochondrial disease. In fact, it's also known that many traditional anti-epileptic medications could actually increase the cellular stress that is part of mitochondrial disease. And so while there may be benefits to some of those medications, some of those benefits can sometimes be countered by an increase in cell stress. And so I think one of the reasons we are advancing 743 for seizures and mitochondrial disease is because we know that 743 targets those energetic pathways that lead to seizures in patients with mitochondrial disease. And so we know that this is sort of a special situation. You see, patients with mitochondrial disease and seizures are not like other patients with seizures. They're different. And having a therapy that can target the things that go on in the cell that result from mitochondrial disease and cause seizures is a very, we believe, very rational approach and promising approach.
Thanks. Can you talk a little bit about the criteria a little bit more? And a question came in asking what would be some of the, if you have the genetic diagnosis, the confirmation, you're having the seizures, you're meeting the other criteria that you talk, what could be certain criterias that might disclude a participant from the trial?
So we have crafted this criteria to be as inclusive as possible. So we want to exclude only if it's absolutely necessary to exclude. So here, as we see in this slide, the caregiver not willing to fill out seizure diaries, right? That's our primary endpoint that has to be done. So careful completion of the diaries on a daily fashion is required. So that, for example, is something that is absolutely needed. Not much else really. If they fit the age criteria and the genetic testing criteria, there's really not much that will exclude the patients.
Okay, and can you explain a little bit more about the EGG? And you said it doesn't have to show an active seizure at that time. Can you explain a little bit more about that criteria and that?
Right. So what we want is simply an EEG that is compatible with diagnosis of an epileptic syndrome. That does not mean that we need to catch the seizure. That would be very difficult to catch, a seizure during the moment we do the EEG. But an EEG that is compatible with that diagnosis, it's EEG that has some sort of a background abnormalities that are compatible with a seizure diagnosis, that is sufficient. Does not have to be done even within a specific amount of time. It has to be just a historic EEG that most likely this patient have done at some point during their diagnosis.
Okay, and just one last question, and this is the classification of mitochondrial epilepsy, is that a formal diagnostic term, or is that more of a classification?
It is, it is a way to group a number of conditions that have in common a genetic mistake that within the mitochondrial genes, either in nuclear or mitochondrial, and also a concomitant disease and a concomitant seizure condition. So it is, if you want, a syndrome that we group as that we create, the group that we create that has these concomitant situations, these concomitant defects.
Yeah, and I would also add that I think everyone in the community is familiar with the term mitochondrial myopathy, right? And you had clinical trials in myopathy, which
Right.
is muscle problems particular to people who have mitochondrial disease. And mitochondrial epilepsy is very similar, right? It's that special condition of seizures in patients who have mitochondrial disease. And as I said a few minutes ago, we know that patients with mitochondrial disease have seizures that are different than other patients who have seizures. We know they result from different things. They result from what, as Francesco said, from genetic defects in the mitochondria. And so it really is a way to appreciate and as Francesco said, collect really a problem that's really significant for people with mitochondrial disease that has its unique source, and that's the mitochondrial genetic defect. I do believe it's going to also be included in an upcoming something called ICD-11, which is the International Classification of Disease, which is a manual that doctors and healthcare providers and insurance companies and the government uses to recognize and give a special number to every possible diagnosis you could think of. And I believe in the upcoming version that's going to be released in the next couple of years, mitochondrial epilepsy is there. Really a formal appreciation of what we know to be a particularly important element of mitochondrial disease.
Right. And that, I mean, and that's a huge step for mitochondrial disease in general, to have those classifications, especially on ICD, because it gives validity to it, right? Because you go in and have these symptoms and it gets coded as something else. And so mitochondrial disease falls off the map. And that will be huge.
And I think, Kyra, that's a credit to you and MitoAction and the work that you do and the patient community does in the incredibly important efforts of educating not only physicians but also payers and the government. That advocacy work that you do is so important to gain recognition for mitochondrial disease and people to understand its complexity and the fact that there is something called mitochondrial myopathy. There is something called mitochondrial epilepsy, and it's different. And it's different because of the unique nature of mitochondrial disease. So that's a success that Kyra, you, MitoAction, and the other patient advocacy groups really deserve a lot of credit for.
Thanks, Matt. I appreciate that. So that was our last question. I can't thank you both enough for your time today to share about the PTC743. I mean, this study opens the door to such hope for this community. And I think, Matt, especially you've been working so long on this therapy, and now along with Dr. Bibbiani and the team at PC Therapeutics, your tireless efforts to bring our community a potential treatment is appreciated more than you will ever know. You guys are truly champions for the mitochondrial disease community, and we thank you truly for all that you're doing. And we're really excited to learn more. I would encourage anyone who is interested in sharing this information with your clinician and to learn more about enrollment is to go to clinicaltrials.gov. And we also have information about the trial on the MitoAction website, or you can check the PTC Therapeutics website as well to learn more. So thank you both for your time.
Thank you very much.
Thank you so much.
So just as a reminder, today's presentation will be posted on the MitoAction website for anyone who would like to listen again, share with others, or go back to and listen at a later date. And you can also find the full catalog of our expert series presentations on Apple Podcasts, Google Play, and Spotify. So thank you everyone for joining us today for today's monthly Mito Expert Series. Be safe, and if there's anything MitoAction can do to support you and your families, please don't hesitate to reach out to us. So enjoy the rest of your day. Have a great weekend, and we look forward to you joining us again next time. Thanks so much, everyone.